So I want to start out with this image and see what you guys think about it. See what kinds of things you notice. And you should have read about necrosis, apoptosis, cell injury, cell death, and we're going to get into the morphology of these things today. So when you see up on here, and I already gave you a big clue that this is the liver, what kinds of things do you notice in this? Yes? The clear vacuoles. So I think you're like this and this here? Right. Do you know what that is?
I do, but I forgot.
Does anybody know what these vacuoles? Uh-oh. So we're looking here at these vacuoles, these large, they're fat-filled endosomes. That's fatty change. It's something that happens with cell injury, specifically in tissue types that metabolize a lot of fat, such as the liver. So that's one thing. I saw some other hands. What else? Yes? Glycogen? Exactly. Yeah, so the liver is a storage organelle, and it does store large quantities of glycogen, and sometimes it's hard to see that here. Sometimes you may see that. The ones that are really pronounced are the ones that are full of triglycerides, the fat ones, but you do store a lot of glycogen as well. What other things do you notice on this image? Do you think the whole section is affected? Yeah.
The left of it is on necrosis of the liver tissue.
Yeah, so over here, on this side, there's a lot more damage. Whereas over here, you're seeing more of the normal liver architecture. And if you notice, these little really dense dots, those are nuclei. Those are pycnotic nuclei. So we'll get into all of this, and you should be able to recognize signs of cell injury and cell death by the end of this lecture. So far, we've talked about injury, and we talked about adaptation. Today, we're going to move from injury into cell death. So here's our outline. I'm going to briefly review the mechanisms of cell injury that we talked about at the end of class last week, and then I'll talk about the morphology of cell injury. What are the changes that you expect to see under the microscope in cells that have undergone some sort of damage? Cells that have undergone irreversible damage are destined for death. So we'll talk about cell death and what you would see with cells that are undergoing necrosis, as well as apoptosis, which is a specialized form of cell death. And I want you to be able to compare those two. And then finally, we're going to talk about the morphologic patterns of necrosis. If we don't get to that today, we'll start off with that on Thursday. So this is our map. This is kind of the map of Chapter 1, Cell Injury and Cell Death. And we started out talking about homeostasis and how cells stay within their physiologic parameters through different mechanisms. If they're exposed to chronic stress, they have ways of adapting, and different tissues will adapt in different ways. If they're exposed to an acute stress or a really severe stress, that's an injury, and we undergo cell injury. And we talked about the mechanisms of those cell injury. So what happens to these injured cells? Well, they're going to have some subcellular alterations related to that injury. And if I were doing this, I would draw a little arrow here, because sometimes those alterations are actually adaptations, and the cell can continue to function in those adaptations, like the fatty liver that we just saw. Reversible cell injury are changes that are characterized by some of these alterations and by other changes But if the stress, if that injurious stimulus is removed, this cell can still go back to normal. If the stress is so severe, or if it lasts too long, you go into irreversible injury and that cell is going to die. There's two ways that a cell could be activated to die. It can be injured so badly that it can't recover, that's over here, or some of the injury, some form of injury will activate caspase proteins, and those caspase proteins start a series of biochemical events in the cell that lead to programmed cell death, which is called apoptosis. So quickly, let's review the mechanisms of cell injury that are going to then lead to cell death. And remember, we talked about the causes of cell injury. We talked about hypoxia, which is a lack of oxygen, and that's a really important one because all cells need oxygen to produce ATP. ATP is what drives cell functions. But then we have all of these other causes of cell injury too. And I asked you at the end of last lecture to go home and when you were studying this material to create a concept map. How many of you actually did that? We started them in class, not too many, a few people. And maybe you don't know what a concept map is, or maybe you haven't done it yourself before. So I just wanted to put something together here. I'm going to show you my concept map. And I want you to compare it to your understanding of the material. And let's see how it turns out. So when we're talking about cell injury, that's our main concept. So I put that right here in the middle. And then remember, I gave you 4 alterations to the cell, 4 types of cell damage that lead to cell injury, that cause that cell to be injured. And those were protein damage, DNA damage, mitochondrial damage, and membrane damage. And these are all interconnected through the biochemical pathways that I mentioned: decreased ATP, increased intracellular calcium, and a production of reactive oxygen species. So which of these leads to decreased ATP? Which of these 4 types of cell damage? Yes?
Mitochondrial damage?
Right. So mitochondrial damage is going to lead to a decrease in cellular ATP. And that's going to have a lot of other effects on the cell because ATP is used for a lot of ATP-dependent proteins and enzymes. So we're going to have a decrease in enzyme function. You know that a lot of the enzymes in your cells, like the sodium-potassium ATPase, that's the enzyme in the cell membrane that pumps sodium and potassium across the membrane to keep the cell in the right osmolar balance. So decreased enzyme function, that's going to be one cause of protein damage. So that's how we get from mitochondrial damage to protein damage. If you're decreasing the ability to maintain that ion gradient, that's also going to lead to membrane damage. Because these ion channels are no longer functioning properly. Decreased enzyme function. Enzymes are really important for repairing DNA. So we can get DNA damage by not having our enzymes work correctly. So you know that DNA polymerase, RNA polymerase, all of your restriction enzymes that are looking for DNA breaks and trying to make repairs. So by a loss of DNA repair, we're going to get DNA damage, all from this. Remember, another byproduct of decreased ATP is that cells that have the ability are going to switch over to fermentation, and we're going to get increased lactic acid. Cells don't like acid. So that acid is also going to affect enzyme function. It's going to cause protein damage because proteins denature in an acidic environment. So that's one of those biochemical pathways. And there are lots more arrows I could draw on here. So let's change over to another one. What's another one? Let's stick with mitochondrial damage for now. What else happens if you have mitochondrial damage? Which of those three? Yes?
Don't you have cytosome C?
Right, so you can get the cytochrome C oxidase, and you can activate the caspase pathways. And so mitochondrial damage is actually going to end up leading to apoptosis through caspases in some circumstances. Also, if you have mitochondrial damage, remember mitochondria is where you're using oxygen. And so anything that's disrupting your normal oxidative phosphorylation can end up leading to increased reactive oxygen species. And reactive oxygen species are free radicals. These are things that have an extra electron, or they're missing an electron, and so they're really reactive with every other kind of molecule around because they need that electron. And so they can directly cause damage to the membrane. So they can knock electrons off from those phospholipids in the membrane. They can directly cause damage to DNA, and they can cause damage to proteins. So the reactive oxygen species are like little bombs inside the cell. They can also, cause further damage to the mitochondria. So we're going right back up here. So reactive oxygen species affect all of these aspects of the cell. Another mechanism was the increased intracellular calcium. So if we have membrane damage, let's say our pumps aren't working correctly, our sodium channels open, our calcium channels open, we can lose that ion homeostasis, and we're going to end up with an influx of intracellular calcium. And that's bad. Ooh, that green doesn't show up too well. Let's try this. So you have an increase in intracellular calcium that can occur when you have membrane damage. That increased intracellular calcium is going to cause the cell to swell because water is going to come in with it. That can cause further membrane damage. And then the main thing that calcium does, it's used by the body as a signaling activator. So most of your enzymes, or many of your enzymes, are activated by the presence of calcium. Normally, intracellular calcium levels are really low, so that you can tweak it. You can let just a little bit of calcium in and start activating proteins, and that is a form of cell signaling. So what that's going to do is it's going to activate a whole bunch of enzymes that shouldn't be activated. So some of these are endonucleases, and they're going to cause DNA damage. Some of these are proteases, and they're going to cause protein damage. And some of these are phospholipases, so they're going to further cause membrane damage or mitochondrial damage. Now you see, that's kind of a mess, right? This concept map is not about the end product. It's really about the process, because as we're trying to connect everything, those ideas are connecting in your mind. You're trying to see how things are related. And even if you get this end process, if you were to give this to somebody else and say, see, I understand cell injury, they'll be like, whoa. But you do, you understand it because you've made all of these connections. It's really hard for somebody else to take your concept map and see all of those connections because it ends up just a jumbled mess. So I really encourage you throughout the semester to do this type of thing regularly to help you see how all of the ideas that we're talking about in class are interrelated. I want to take just a second to go back to reactive oxygen species, because I had a few questions about them, and this is a really important mechanism, and it's becoming increasingly active area of research. So those of you who are interested in research, this is something to consider. So reactive oxygen species are also termed free radicals, and the media has picked up that term, so you've probably all heard of free radicals. So these are molecules that have an extra electron. And you can see here, or an unpaired electron, I should say. So our normal oxygen, remember in the outer valence, in the second ring, if you're not hydrogen, you want 8 electrons. Oxygen has 7, and so it binds to another oxygen molecule to share those electrons, and you get that double bond, and that's a nice, happy molecule. But oxygen itself is pretty reactive, and so sometimes another electron will jump on there. And so that will form peroxide. This is unstable because there's too many electrons in the outer shell, and those electrons, those extra electrons, are unpaired right there. If only one jumps on, it's called a superoxide anion. You can get a hydroxyl radical. Have you guys talked about this in organic chemistry? Have you done a little bit about this? Some yes, a little bit. Or maybe in biochemistry. I encourage you to be thinking about this if you are currently taking biochemistry or if you're going to take it in the future, because it's really interesting and it makes the chemistry make a lot more sense. At least it does for me. So these are what I mean by ROS, reactive oxygen species. These are molecules that have oxygen and that are unstable because they have unpaired electrons or extra electrons. Like here, the hydroxyl ion has extra electrons. And so what happens inside the mitochondria, normal oxygen will come in, and then if the mitochondria isn't functioning correctly, or if there's too much oxygen, then you can get incomplete oxidative phosphorylation, and that produces some of these free radicals or reactive oxygen species. So you get a lot of this generation of superoxide. But your body has a mechanism to take care of that. And so one of the enzymes that's really important is called superoxide dismutase. Also known as SOD. And so what that does is it takes this superoxide and it reduces it to hydrogen peroxide, which is still not a great molecule to have around, but it's a little bit safer than the superoxide. So superoxide dismutase, this enzyme, is actually found to be genetically altered. It's mutated in people who have familial ALS, amyotrophic lateral sclerosis, which is a neurodegenerative disease of the motor neurons. And so they have progressive muscle weakness, they can't move, and they eventually die of respiratory failure. And there are about 10% of cases of ALS are linked to a mutation, a genetic mutation in this enzyme. So this is the accumulation of free radicals inside those motor neurons that ends up causing death of those neurons, and it leads to paralysis of the body. Yes?
Okay, so like what exactly is the point for superoxide to get changed into hydrogen peroxide? Is it just for our bodies to, I don't know, get used to it or like take care
Yeah, so what is the point of changing superoxide into hydrogen peroxide? It's simply to get rid of this free radical. So free radicals are a natural byproduct of using oxygen to create energy. So we all create free radicals at low levels just because we're doing aerobic metabolism. And because we want to push ourselves, anytime you undergo exercise, for example, you increase that production of free radicals because you're increasing the aerobic metabolism, you're bringing more oxygen into the cells, and so you get even more generation of free radicals. And so evolutionarily, we've been able to deal with that by having enzymes that can reduce those free radicals. So all of our cells in our body have some version of superoxide dismutase to take care of this process. And like I said, hydrogen peroxide, as you guys know, it's not that clean of a molecule. It's still somewhat reactive, and it can quickly break down into these hydroxyl radicals. So these hydroxyl radicals are also swimming around in your mitochondria inside your cells, ready to cause a lot of damage. And so another protein we have, another enzyme, is called glutathione, which maybe you've heard of. It's, we have this in the cytoplasm, in the mitochondrial membrane of a lot of different cell types. And it reduces hydroxyl radicals so that you don't have these free radicals swimming around throughout your cells. One of the places that you find this a lot in high quantities is the liver. And that's because one of the liver's jobs is to metabolize things that you ingest. So a lot of chemicals are metabolized inside the liver. So let's go back to our image of liver toxicity here. This is actually a histology slide from a person who overdosed on acetaminophen. So you all know that Tylenol can cause liver damage, right? You've heard that? And it's not uncommon to have people take high doses of Tylenol and end up with something like this, hepatic failure. And what happens is that when Tylenol gets into the body, it goes into the liver, and the cells of the liver start to metabolize it. And one of the breakdown products of acetaminophen, through this metabolism by the liver cells, actually reduces glutathione. And so this glutathione gets used up in the liver in a person who's had too much acetaminophen, and then it's not there to combat free radicals. So the glutathione gets used up, free radicals have their heyday, and they cause necrosis of the cells of the liver. So that's what's actually going on in acetaminophen toxicity. That's why the safe dose, if you just take 2 capsules, if you take 400 milligrams, your liver can keep up. So you don't generate so much reduced glutathione that you can't combat the free radicals. You can keep turning it over and have enough to keep up. It's only if you have large quantities all at once that you overwhelm that mechanism for dealing with free radicals and end up with tissue necrosis. So I wanted to point out Tina Trustador in the biology department does a lot of research on oxidative stress. So right here at NAU, we are conducting research in this field. And you can see this first article. She's been looking at the generation of free radicals, oxidative stress, in acute exercise. She also is very interested in aging. So it appears that our ability to combat free radicals diminishes with aging. And so a lot of her research is focused on the ability of continuous exercise or dietary components, such as tart cherry juice, to help overcome this natural decline of free radical mechanisms in aging. So I encourage you, if you're interested in this, we have research going on here. Go ahead and find out more about it. There's a lot of research about it. And it also comes back to this idea of hormesis that we talked about last time. Is everybody clear on hormesis? So hormesis, you have some stress, and that actually turns out to be beneficial to the cells or to the body. So the one I wanted to point out right here, because I didn't get a chance to look at these examples with you, is down here. So looking at this graph, what they did is they exposed mice to gamma rays. Gamma rays are a form of radiation. They do 2 things. They cause DNA damage, and they produce reactive oxygen species, because these radiation waves knock electrons off from molecules. So if you give rats exposure to gamma, all of us would immediately think that's bad. But it turns out at low doses, so right here, so this is increasing dose, at the lowest doses, you actually get a decreased tumor incidence. And then it's only as you get to the higher doses that your rate of tumors starts to go up. So what do you think could be going on? Why would you get this? Yeah. Yep.
It's more like acting kind of like a vaccine. The cells are adapting to.
Yeah, I like that analogy. She said that it's kind of like a vaccine. So your cells are getting a small dose, and they say, you know what? There's some stressors out there. And so what's probably happening is that from a little bit of exposure, you actually increase your DNA repair mechanisms. So you can turn on gene expression and start to produce more of those proteins that recognize and fix DNA damage. And you probably increase these enzymes like SOD and glutathione, which I abbreviated GSH there. So small exposures to stress help your cells deal with that stress better. I just think that's a really cool concept. Okay, so we've gone back over now all of these mechanisms of cell injury. What happens to these cells that are injured? Well, it depends. If the cell has enough time to upregulate some of these protective mechanisms, then, and if the damage hasn't been so extensive, then you can actually get full resolution. So the cell will take a hit, it'll repair itself, and it'll go back to its normal function. That's if you only get reversible injury. As I mentioned at the very beginning, some of these responses to injury actually result in adaptation. So the cell takes a hit, it adapts, it gets stronger, and so it actually changes in response to this, and now it's a little bit better suited to the environment that has that type of stress. If the damage is too great, you can end up with cell death. And then I'm not going to talk about anymore today this idea of cancer, but many forms of cell injury cause DNA mutations that are going to lead that cell down the path of cancer. So these two forms are reversible. Cell death, or if the cell is mutated to the point of turning it into a cancer cell, it can't go back from that. So these two are irreversible. Any questions? All right, so let's take a look at some of these changes that are going on with injury and death. And I first want to compare what you might see in a cell that's undergone reversible injury versus signs that the cell has crossed that threshold and is now irreversibly injured, meaning it can't go back, it's going to die. So what are the things you might see with reversible cell injury? And you can kind of see here, this cell is swollen. Right, so if we have that loss of ion homeostasis, the cells are going to start to swell. Part of membrane damage, some of that membrane is going to get irregular. So if you see the shape of the cell doesn't look normal, it's kind of got these little blebs on it, it's an irregular shape, that's a sign of cell damage. Other things you might see are the individual organelles, endoplasmic reticulum, the mitochondrium. Those also will be swollen. So swelling of the cell and the internal organelles. And then one thing that is not shown here is fatty change. So what we saw in some of those liver cells that were on the non-necrosis side of liver toxicity, with those big vacuoles full of lipids, that's fatty change. Those cells are not yet destined to die. They could still go back if the acute injury is removed in time. So fatty change is a sign of reversible cell injury. If a cell sustains too much damage, you're going to start to see down here, these organelles actually start to break down. So the breakdown of organelles is going to appear as a kind of a moth-eaten appearance. So when you see the cell, it looks like it's been kind of little holes taken out of it. And that's what's happening is the organelles are breaking down inside the cell, and they're starting to kind of clump together inside the cell. Really important, with reversible cell injury, you don't see many signs of nuclear changes. So the nucleus is still pretty much intact. You may see a little bit of clumping of chromatin. Chromatin is one of the proteins that the DNA is wrapped around, is structurally supporting the DNA, but you don't see clumping of DNA, you don't see the nucleus starting to change the way you do in irreversible cell injury. So once the nucleus starts to break down, that cell's in bad shape. That cell is going to die. The nucleus is really important. And so some of the nuclear changes you might see are pycnosis. What was pycnosis? Shrinking of the nucleus. So the nucleus starts to condense. And like we saw in that liver slide, you see these really dark, dense, small nuclei in cells that are undergoing necrosis. Karyorrhexis and karyolysis. Do you guys remember those terms from the reading? So what's karyorrhexis? That's fragmentation of the nucleus. So the nucleus starts to break apart. And karyorrhexis is where the DNA is being degraded. And so the nucleus on histology actually starts to fade. There's nothing left for the hematoxylin and eosin to bind to, so that the basophilia, that dark staining of the nucleus, fades away. So that's karyolysis. And you can see any of those types of nuclear changes at different points as the cell is undergoing necrosis. So you get nuclear changes there. The membrane becomes even more irregular and sometimes splotchy. So the membrane starts to break apart. And it's actually spilling its contents out into the extracellular environment. And then part of the changes that you'll see, I'm going to point out when we get to the slides here on histology. So this is an image of the kidney. Can everybody see it? Do you want me to turn the lights down anymore? This is okay for you? Because I don't want you guys to fall asleep, but I want to be able to point out what's going on. So A, this is a normal liver. So these are the tubular cells, And you can see they have a nice uniform appearance. So the cells that you don't see a nucleus in, it's just because of the plane of the image. These are thin slices through the kidney. But you can see the cells seem to be roughly cuboid. They have a nice big circular nucleus in there. And there's no space between the cells. So this is normal. Over here, this is a kidney that's undergone reversible injury. So what are the things you see in these cells? What's that? Swelling. Yes, some of these cells are swollen. So this cell right here looks much bigger than what you would normally expect to see in a renal tubular cell. So you see swelling. What else do you see? Yeah, over here.
The nuclei are still intact.
The nuclei are still intact, so that's important, right? Because that tells us that these changes haven't yet affected the nucleus. So most of these nuclei are still round. They still look very much like the nuclei in the first image. Good. Yeah?
Irregular membranes?
Yes. So look here. Look at these membranes. They're just kind of, they don't have that same intact shape. And what do you notice about the cytoplasm? I haven't mentioned this yet.
It's kind of like irregularly shaped. It has like excess material in it.
Yeah, so it's a little bit irregular, and that's reflecting the irregular shape of the cells as the membrane loses its function. And you can kind of see, I hope this is obvious, see how this cell right here is a much denser pink than these cells over here, or even the one next to it. And that's called, not hemo, sorry. That's called eosinophilia. So what that means is that there's been a decrease in RNA as our DNA is our enzymes aren't functioning, so we're not undergoing protein synthesis, we're not undergoing RNA synthesis. So there's less RNA around, and you end up getting denatured proteins that bind to that eosin on an H&E stain and kind of cause it to clump together. So eosinophilia. Doesn't mean the cell is irreversibly damaged, but it's a sign that things are not going in the right direction. Now, if you move over to the next one, this is irreversible. So these cells in this kidney are undergoing necrosis. And you can see several signs of this. So what do you notice about what this arrow is pointing to? What do you think that is? I hear karyolysis. Did somebody say that? Yeah? So notice how this nucleus right here is faint compared to these over here. And it's actually a little bit swollen. So that's karyolysis. That's the DNA being broken down inside the nucleus. And it's not holding that dye as well. So it's the loss of basophilia. And you can compare that to some of the other nuclei in this image that are actually undergoing pycnosis. And that's where the DNA is clumping together as it sustains damage. And you can a little bit see in this one right here, this is starting to undergo karyorexis. And that was the fragmentation of the nuclear contents. Can you guys see those changes that are going on there? What else do you notice? Yeah,
There's just a lot of open space.
a lot of open space. What do you think that is?
Like this leakiness of like the cytoplasm
Mm-hmm.
is causing a response. Is this inflammation response or is this?
So yes, you do get inflammation with necrosis. And the white space isn't really, inflammation is actually usually seen more as cells coming into the area, but all this white space is indicative of swelling of the tissue itself. So this is where fluid was instead of the cells. So as the cells break down, and as you were saying, as their membrane contents spill out, you're now bringing more fluid into the tissue around it. So you see swelling, not just of the cells themselves, you do see swelling of the individual cells, but you also see swelling in the tissue where those cells are dying. Inflammation can also contribute to that swelling because of the release of cytokines, and we're going to get into that in the next lecture. But you can see in this image now, these cells are huge. So compared to these, or even these injured cells here, the cells that are starting to undergo necrosis are even more swollen. The membranes, look at this cell. You can barely tell that there was a cell there because the membrane is so degraded. And so when you see kind of these really irregularly shaped cells with no nucleus, a lot of these cells have already completely lost the nucleus, then you know that that tissue is undergoing necrosis. It's sustained irreversible injury. Any questions? Yeah.
Would you also see loss of appearance?
Yes, good. So you have, let me move this up a little. You have decreased cell-to-cell contacts. So loss of adherence between the cells. As the proteins get broken down, protein degradation because of those biochemical mechanisms, some of those proteins anchor the membrane to the cytoplasm, and some of those cells anchor that membrane to the next cell over. And so you lose both of those connections. That's part of why the membrane loses its integrity, and then the cells start to drift apart from each other. And you can definitely see that occurring here. You've lost the nice tubular shape of these cells. They're supposed to form a ring. You're no longer getting that over here, and that's because of the loss of cell-cell contacts. Great. Yes?
So would that be a definite trait of irreversible injury, the loss of cell-to-cell contact?
Yes.
Okay.
So if you see loss of cell-to-cell contact, that means the membrane damage is so severe that you've gone over to necrosis. Yes.
What kind of necrosis is this?
Good question. What kind of necrosis is this? Do you have a guess?
I mean, it looks like it's turning to liquid, but I don't really know.
So it does look a little bit like, she said it looks like it's turning to liquid, and what kind of necrosis would that be? Liquefactive necrosis. In fact, kidney usually undergoes coagulative necrosis. So this is coagulative necrosis, and it's a little bit hard to see at this power. If we were to zoom out, you'd see that there's still stuff there. Liquefactive necrosis really just turns into a pool of pus. And so we'll see that, and it's easier to tell on gross in just a little bit. I like that connecting concepts. Here's another image of cell injury. So at the top, this is the normal liver. And you remember those zones of the liver? And here's the triad. So this is normal. This is a liver that's undergone fatty change. You see this with metabolic syndrome and obesity. And you also see it in alcohol damage. So alcoholic liver disease. And with metabolic syndrome, it's becoming hugely prevalent. What do you think you would see if you were to actually look at this liver grossly? What do you think the liver would look like? Yeah, it looks fatty. It's enlarged. So people who have fatty liver actually have increased abdominal pressure. It makes it harder to breathe. They get a lot of indigestion, a lot of stomach pains. And they're also, the liver texture, the consistency of it becomes squishier. It actually, I have a cadaver. Those of you who are doing this as an honors will get to see this. When you touch the liver of one that has fatty liver disease, you can kind of feel that change that has occurred inside the cells. It's really cool. So these vacuoles that are full of fat, if a person has this and they stop drinking alcohol, or they start losing weight and eating a healthier diet, this is still reversible. This will go back. Those triglycerides will get metabolized. New ones won't be replacing it. And so this liver will can still go back to normal. What you saw with liver toxicity, and you can kind of see the difference here from this, where the cells are still intact. Notice that the cells are still next to each other. They just have these big fat globs in them. And that's different than the cells that were undergoing necrosis, where you couldn't even see that there were individual cells there anymore. So another sign of damage that you might see in cells are accumulations of different things. And I just put 2 examples here. So in this image, you see all these little kind of rust-colored dots. That's iron. So this is hemosiderin. So this often will happen in tissue like the liver, the spleen, and the bone marrow, in response to iron overload. Normally, your body's pretty good at regulating intake of iron, but you don't have a good mechanism for getting rid of iron. Bleeding is the main way that you can get rid of iron, by getting rid of extra red blood cells that carry iron in the form of hemoglobin. And so there are people who have impaired mechanisms for regulating iron intake, and they actually accumulate a lot of iron, and it can stick around in the cells, and that's a sign of cell injury. Another one over here, this is lipofuscin. So you can see these are a similar color. You see these kind of clumps accumulating in the cell, and they tend to accumulate around the nucleus. Lipofuscin is still being investigated. It's not completely well understood what's going on, but it's thought that this is a sign of long-term oxidative damage. So you have your reactive oxygen species in cells that aren't able to combat it very well. Those kind of break down some of the intracellular organelles. And as those organelles break down, they clump together and clump onto the nucleus of the cell. So these clumps of lipofuscin are really lipids and proteins kind of stuck together around there. But why this is occurring, when it's occurring, is still really being worked out. We don't know that much, but it does seem to be a sign of oxidative stress damaged from reactive oxygen species, and it increases with aging. So it is another sign of cell injury. And then some other ones, carbon. In people who are smokers or who work in coal mines, and you get a lot of dust in the lungs, the macrophages will ingest those carbon particles or other particles, but then they just hang out in the macrophages. And so when you look at the image, if you look at a slice, or even just grossly, if you look at the lung of somebody who has smoked for a long time or been in a hazardous working environment, they call it black lung. You can actually see this, and the macrophages just sit there with accumulating all of this carbon. Melanin can accumulate in cells. Did you have a question?
Yes, for filters that have carbon in them, if the carbon comes out of the filter and into the water, would that cause damage?
So you mean if you have like drinking water that you're filtering
Yeah.
to get the carbon out of it? I don't know.
Well, don't they use activated carbon?
They do. Activated carbon could be a filter, yes. So you're saying if you drink the water that has the carbon in it?
Yeah, like some of the dust from the filter comes out into the water.
Yeah, that will, if you drink, so it won't go into your lungs because you're drinking it. Hopefully, that'll just pass through you. If you don't have a lot of damaged cells that activated, if you were to ingest activated carbon, it would just pass through. It's really if you breathe it in, it kind of sits around there because those cells, it doesn't have a good mechanism for getting out of your lungs. Does that answer it?
Yeah.
Kind of? And then one more type of accumulation is calcium can accumulate. And calcium accumulation is usually extracellular, so this isn't inside the cell. It's more a sign of tissue damage than actual cell damage. But it tends to happen in areas that are in tissue where necrosis is happening. So dystrophic calcification is happening in necrotic tissue. And what you see here is the tricuspid valve of the heart. So this is an image from looking at the heart, and you're looking down at the valve. And all of this white stuff are calcium deposits. So as the cells undergo damage, they leak out their cytoplasmic contents, and some of those form salts with calcium that's in your extracellular space. And so you get the deposition of calcium salts. Once that calcium has become a salt, it tends to be more insoluble and it kind of lays down right there on the tissue. When we talk about atherosclerosis, you'll see that this is an important mechanism in the process of atherosclerosis as well. In this person, they probably had some kind of damage to their valve. It can be caused by increased blood flow, increased work of that valve against high blood pressure. There are several causes that can lead to valve damage. Also, congenital problems can lead to this formation of dystrophic calcification. So dystrophic calcification is pathologic, and it's happening when the tissue around it is being injured. And that's different than metastatic calcification. So metastatic calcification, you might see the same thing. It won't look that much different, other than it occurs in areas where there is no tissue damage. So why would you get calcification where there is no tissue damage? Yeah. Hypercalcemia, exactly. If there's too much calcium in the extracellular environment, it'll start to just deposit. And it'll just deposit wherever it can find a spot. It doesn't have to be damaged tissue. So and there's a lot of ways you can get hypercalcemia. Parathyroid tumors, kidney damage. You can get it, a lot of tumors like carcinoid tumor of the lung will actually secrete parathyroid hormone, and that causes increased calcium levels in the blood. If you have cancer and you're actively undergoing chemotherapy, as the cancer cells die, they release a lot of calcium into the blood. And so you can get hypercalcemia associated with treatment of cancer, actively dying cancer cells. All right, we got to the break. So let's take a 2-minute break, and then we'll come back and we'll do patterns of necrosis. Yes?
Would it be B?
You think B is necrosis? How many people agree that B looks like necrosis? All right. What are your clues? What are you looking at there? All the holes in it. So you're seeing all of these holes, and this is an electron micrograph. So we're not actually looking inside the cell, we're looking at the cell surface. So all of these holes are actually membrane defects. Your membrane is breaking apart here. So whatever's inside the cell can leak out through these holes. What else?
It looks like it's shrunken on the right side, and it's not as swollen as the left side is.
Right. So the one in A doesn't look as swollen as the one in B. So this one is actually starting to kind of condense down, and this one is, if they look roughly the same size to me, that's probably because a lot of the cytoplasmic contents have leaked out. If you were to see it a little bit earlier, it would be swollen compared to a cell that undergoes apoptosis. Notice the membrane over here, how irregular it is. So you have holes, you have irregular membrane. And then if we were to look inside, the nucleus would of course be starting to break down in the necrotic cell. What do you think this is? Because this is a membrane change. Why is this not necrosis? So Alex, you're saying this is blebbing? Right. So this is, and blebbing's a weird term. It's a histologic term or a pathology term, but it can be used to describe both. Some people will call these little blebs. That's why I say membrane irregularity. This, you could call it blebbing or budding, but it's actually part of the cell breaking off, enclosed within its membrane. So that's an important point. The membrane stays intact here in apoptosis. And does anybody know what this would be called once it kind of breaks off from the main cell? Carl, don't give away the answer. What would it be called? Vesicle. So the vesicles are actually inside the cell still. So as the cell starts to break down through apoptosis, these are called apoptotic bodies. And we'll see a cartoon of that in just a second to help illustrate the point. But I want you guys to take a few minutes from your reading, if you took notes or outlined the chapter, or if you have your book here, work with 2 or 3 other people nearby to try to fill out this chart. I want you to compare necrosis with apoptosis. Yes, question.
So on the quiz, there was a question about which ones included blebbing. So would that be apoptosis?
I have to go back and look. Look at the actual question. There was a little bit more to it, right? Yeah. Let me go back and check on it. So that's why I don't like the term blebbing. Blebs are basically just little outpouches of the membrane. So you can see them in apoptotic bodies, which are huge blebs, or you can see membrane irregularities, which are like little blebs. So I'll go back and look at that question to make sure that we're all clear on terminology there. Okay, I'm going to come here to the back to get some some feedback on this. Turn my thing on. All right. So back here, what happens to cell size in necrosis? I heard it, swelling. So cell size is going to increase. What happens in apoptosis? It's going to decrease. Right, the cells in apoptosis, those caspases, once they're activated, they start telling the cell to go ahead and clean up. So it breaks down proteins, it packages them into nice little vesicles. It starts telling the membrane to pinch off, bud off small apoptotic bodies. The whole cell shrinks down. It shuts down production of ATP, and the cell quietly disappears. What happens to the nucleus in necrosis? Somebody over here. What do you guys think? Pycnosis, karyolysis. And what else? And karyorexis, yes. What about in apoptosis? What happens?
Shrinks and breaks off all of the DNA into tiny.
So, like the whole cell body, the nucleus in apoptosis is going to fragment into small pieces. Remember, karyorexis also means fragmentation. So, fragmentation alone doesn't help you differentiate between the two. But if you see pycnosis or karyolysis, that's definitely necrosis and not apoptosis. How about membrane? What happens with necrosis? We saw that on the electron micrograph. Yeah, it breaks down, it forms holes. Whereas in apoptosis, very important, the membrane stays intact. So you have those apoptotic bodies coming off, but the membrane itself stays intact. Yeah, question.
Question about the nucleus of apoptosis. On the quiz, it said it fragments into larger fragments.
Yeah, I'm going to go back and look at the quiz. It sounds like the wording in that was confusing. Somebody else asked as well. So I will send out an announcement. I'm going to go back and check. I can't remember what the wording on those was. But this is important to know those differences between the membrane changes that you'd see, because that's the main difference between apoptosis and necrosis, are those membrane changes. And that membrane stays intact. What happens with inflammation? Do you get inflammation with necrosis? Yes. Do you get inflammation with apoptosis? No. And it's because the membrane stays intact. So if the cytoplasmic contents leak out, that can trigger inflammation because you have proteins and you have molecules where they're not supposed to be, and that triggers the whole inflammatory process. Whereas with apoptosis, everything stays contained. So it is macrophages. You do have those phagocytic cells that are eating up those apoptotic bodies, but it doesn't trigger all of the other steps of inflammation. So that's just normal cleanup that happens. And then what did you guys come up with as the main stimulus for necrosis? I heard something. Pathologic. Necrosis is always pathologic. And so when we were talking about those mechanisms before, any of those mechanisms that cause irreversible cell damage can lead to necrosis. So it is nonspecific cell injury again, I want to stress this idea that I showed you with all of those arrows going all over the place, pretty much any of those biochemical events that lead to membrane damage. So the mechanism is membrane damage. But as you saw, you can get membrane damage because of hypoxia with decreased ATP production. You can get membrane damaged with reactive oxygen species. You can get membrane damage with increased intracellular calcium. So there's a lot of pathways that can lead to that. But ultimately, it's when the membrane starts to break down that you start with necrosis. Versus apoptosis, the stimulus for apoptosis are pretty specific. There's both physiologic, and these are things like the cells no longer receive trophic support. They lose their hormone support. They lose growth factor support. And so they lose cell contact with their neighbors. So those types of things are a signal to the cell to start to undergo apoptosis. You can also have pathologic, and this usually occurs because you have DNA damage or some forms of mitochondrial damage. And what it really requires is you have to activate these caspase proteins. So caspase proteins are the trigger for apoptosis, and they're activated in specific situations that are outlined in your book. Does everybody understand those differences? I get a feeling we're out of time. All right, I'll see you guys on Thursday. I will look into that question on the quiz and send out an announcement about it.